Now, just a bit of warning, I think you can look at the length of the video and you know that this is going to be a long one. Now, if you remember in the previous video, I told you that in dihybrid inheritance, we are interested in the inheritance of two different genes, and we are talking about the case of unlinked genes. And in unlinked genes, I said that the genes are on separate chromosomes. As an example, I'm using gene for body color. Gene 1 affects body color, where it has the dominant B allele makes it blue, the recessive B allele makes it red, and another gene, which is gene number 2, which affects body shape, which capital T allele, dominant, makes it triangle, small t allele makes it square. Like I said, do not memorize my example. This is just the way I'm teaching it. Now, genes 1 and 2 are unlinked. They will also usually explain that in the exam. Okay, they will say that the gene for body color and the gene for body shape, the two different genes are on different chromosomes or they are unlinked. That is your clue basically in the exam. Or they will usually say it's located on different chromosomes or it is unlinked. Now, in my example here, I'm going to start off the ball rolling by drawing out the 4 chromosomes, where the larger chromosome, where I've highlighted in orange, has gene number 1, which affects body color, and the smaller chromosomes at the bottom over there contains gene number 2, which I have highlighted in green, and that gene number 2 affects body shape. As you can see here, gene number 1 and gene number 2 are on different chromosomes, so they are unlinked. So let's cross organism that is homozygous dominant and homozygous recessive. Now, what does it mean by homozygous dominant? Homozygous dominant means both the alleles are the same, homozygous, and both the alleles are dominant, which means to say the organism for gene number 1, it is large B, and for gene number 2 is large T. For the homozygous recessive organism, homozygous, same alleles, recessive means, the smaller alphabet. So in this case over here, it is small b, small t, as you can see there. When you're asked to write the genotype, when it comes to dihybrid inheritance and unlinked genes, you just have to write the genotype as, for the left organism, large B, large T, and for the organism on the right, small b, small t. Very simple. Okay, so that's how you write the genotype. What's the phenotype of the organism on the left? Because it has the large B allele and large T allele, the dominant alleles are always expressed no matter what, so the organism will be a blue triangle. Simple. But for the homozygous recessive organism, it doesn't have dominant alleles at all. It only has small b alleles and small t alleles. So small b makes it red, small t makes it square. So it's a red square, as you can see here. Now, when they produce gametes, let's look at the organism on the left. Remember, its chromosomes is large B, large T. And usually before meiosis, I told you before, the chromosomes will replicate to form the sister chromatids, right? And then during meiosis 1, what happens? The homologous pairs separate, okay? When the homologous pairs separate, I'm just going to show it to you over here. As you can see here, that set of chromosomes separate into that cell, and that set of chromosomes separate into that cell. And then after that, meiosis 2 happens, and the sister chromatids will separate. And when the sister chromatids basically separate, what happens is they produce the 4 cells. Now, when they produce the 4 cells over here, each cell will have one large chromosome and one small chromosome, as you can see that, okay? What allele does the large chromosome contain? It contains one large B. And what allele does the small chromosome contain? It contains large T. So all the cells, all the gametes over here will always produce large B, large T alleles, no matter what. Now, immediately some students will say, wait a second, I thought meiosis was supposed to produce genetically different cells. It was, but in this case, they only have large B alleles and large T alleles. That's the only information they can pass down. So no matter what in this case, no matter how many gametes it produces, it will always just have one large B allele and one large T allele. And then, of course, for the homozygous recessive one, what's going to happen? I'm going to change the alphabets, as you can see here, small b, small t, and then the gametes will also be small b, small t's, no matter what. So 100% of the gametes are small b, small t. So this is the gametes produced by the homozygous dominant and also the homozygous recessive organisms. Now, what happens to gametes? Gametes will then fuse with each other. When the gametes fuse with each other, what happens then is the organism will receive one set from the parent, which is large B, large T, and another set which contains small b, small t, as you can see over there. So the offspring's genotype is large B, small b, large T, small t. It becomes heterozygous. Heterozygous means the alleles are different. Large B, small b is different. Large T, small t is different as well. And what's the phenotype of this organism? The phenotype of this organism is, because it has one large b and small b, the dominant allele, which is large B, will get expressed. So the organism becomes blue, and because it has one large T and small t, the large T allele, which is dominant, gets expressed over the recessive allele, and therefore the organism becomes a triangle. So the phenotype is blue triangle. 100% of the offsprings produced by these two parents will be blue triangles, no matter what. So let's make it even more complicated. Okay, let's take two heterozygous organisms, large B, large T, small t, bred together with large B, large T, small t. In this case, here is where it becomes complicated. I'm drawing out the chromosomes on the right over there, unlinked 4 chromosomes, large B, large T, small t, as you can see there. So what you notice is I'm coloring one set of the chromosomes in blue and one set of the chromosomes in orange. The blue chromosomes contain one large B and one large T, and the orange chromosomes contain one small b and one small t. Okay, now remember, chromosomes will replicate usually. And if you remember in the previous video, I'm going to put it on the top right corner, I told you that a process known as independent assortment or random assortment can happen. Now, what exactly happens during independent assortment or random assortment? Remember, during metaphase 1, the chromosomes will line up along the equator, and when they line up along the equator, the bivalents will line up along the equator and the spindle fibers attach to the centromeres. But during random assortment, they can spontaneously swap places. As you can see here, I hope you can appreciate this. As you can see, one of the bivalents have swapped places. I'm just highlighting it there. I want you to see those two highlighted parts to see how they are different. Okay, some students will ask me which one will happen more often, the one on the left or the one on the right. Both processes can happen equally. Okay, it's quite unpredictable that way. So why is this such a big deal? Because when the homologous pairs separate during meiosis 1, it will look like that. And when it separates during meiosis 2, the chromosomes, if you look at this one, you can only get small b, small t, small b, small t, large B, large T, large B, large T. But in the case of the independent assortment on the right side, you can get small b, large T, small b, large T, and large B, small t, large B, small t. That is a bit interesting over there to notice, isn't it? So in this case over here, we notice that random assortment can give you different types of gametes. So, because of independent assortment, how many types of gametes can be produced? You can produce 4 types of gametes. You can produce either small b, small t gametes, or large B, large T gametes, or small b, large T gametes, or large B, small t gametes equally. Each gametes have a 25% chance of being produced. The proportion of these gametes are almost equal. Okay, and of course, for the organism on the right, it's also able to produce those 4 types of gametes as well. Here is where it becomes a little bit difficult. How are you going to cross them together? It's not easy to cross them together, right? Because, I mean, it's possible, but there's a very high chance of making mistakes over here. So, to not make mistakes and to make your life easier, we construct something called as a Punnett square. As you can see here, the organism on the left, I'm highlighting the gametes in yellow color for your reference, and for the organism on the right, I'm highlighting the gametes maybe in a green color, as you can see there, and I'm just drawing out the Punnett square. So, okay, so I'm just going to zoom in, and remember, when you're drawing out the Punnett square, you need to put inside the square, you need to put in the offspring's genotype and phenotype. I am going to do the first one for you. So the first box over here, these gametes, where this gamete over here and that gamete over there will fuse with each other, and you'll get large B, large B, large T, large T. Now, I'm going to give you some time. You can pause the video, draw out your own Punnett square, put in all these alphabets, and I want you to correctly fill in all the genotype of the offsprings. All right, and once you're done, unpause it, and you may then see if your answer is correct. So this is the entire thing, as you can see over here, where I've put it in. This is I hope your answer is correct. Now, once you've put in the genotype, then you put in the phenotype. Phenotype is very simple. As long as the organism has a large T, it will be a triangle. So any boxes with a large T in it, I'm just going to put in the triangles first for all of them. As you can see here, as long as there's a large T. Whether it's one large T or two large T does not matter, because capital T is the dominant allele. So there you go. Then after that, as if it doesn't have large T at all, which means if it only has small t, it's recessive, so we just put in the squares. That's it. Okay, so we've put in the squares already, and then let's color it. So when we color it, as long as it has large B alleles, blue in color. Now, I know my coloring sucks over here. I'm not coloring within the line, but don't judge me for this, okay? I think this video was done at 3 o'clock in the morning, so I was really tired. And if it's only containing small b alleles, it will be red in color. There you go. Okay, so I've colored it in. Now, some students will ask, in the exam, do I have to draw out the phenotype? No, write it out. So write out the phenotype as blue triangle, blue square, red triangle, red square. Put in the phenotypes together with the genotypes in the same box. Okay, and then more importantly, when you look at the offspring phenotypes, you can get 4 possible phenotypes in the offsprings. You can either get blue triangles, blue squares, and red squares. So count out what's the possibility of getting a blue triangle, 9, red triangle, 3, blue square, 3, and red square, 1. So in this case here, the ratio of the offsprings will be 9, 3, 3, 1, which means there's a 9/16 possibility to get a blue triangle, 3/16 to get red triangle or blue squares, and 1/16 chance of getting a red square. So in dihybrid inheritance unlinked genes, when 2 heterozygous are bred, the offsprings will usually show a 9:3:3:1 ratio. You need to remember this magic number. It is extremely important. Now, let's try an actual example. So, the fruit color in courgette is controlled by the gene capital A small a. Some students will ask the question, what the hell is a courgette? A courgette is, I guess you can say, a variety of vegetables. Zucchini is an example of a courgette, by the way. If I'm not mistaken, it's a class of vegetables like squash, zucchinis. If I'm not mistaken, pumpkins fall under courgettes as well. I might be wrong, but yeah. Now, the fruit color in courgette is controlled by the gene large A small a, and the fruit shape in the courgette is controlled by the gene large B small b. So you have 2 genes here. You have 1 gene for fruit color and you have 1 gene for fruit shape. Gene A and B occur on genes A and B occur on different chromosomes. That's your clue to tell you that the fruit color gene and the fruit shape gene are unlinked. You must be aware when they mention that in the exam. Now, they said that a yellow fruit is produced when the dominant allele A, large A, is present. So large A makes it yellow. And we know that based on the table, small a makes it green, as I've highlighted over there. So that's your clue. So sometimes you kind of have to piece it out. And the gene for fruit shape, as you can see here, large B, they said that a round fruit is produced when the dominant allele is present. So large B makes it round, small b makes it, small b here makes it long, as I've highlighted over there. So I'm going to give my own twist to this question. The question was a little bit different. I'm going to say that the parent is a heterozygous, okay? And one parent, I've just put the phenotype over there, green and long. Sometimes you as a student will have to piece out the genotype. If it's heterozygous, we know that heterozygous just means the alleles are different. Large A, large B, small b. Simple. And for the green long one, it can only be green and long because of the recessive alleles. Green, which is small a, long, which is small b. So sometimes you might have to piece out what the genotype is. And of course, for the parent, which is heterozygous, it is yellow and round because it has one large A and one large B allele. Okay, remember, I'm just drawing out the chromosomes over here. As you can see, unlinked genes, large A, large B, small b for the parent on the left, small a, small b for the parent on the right. Okay, now if you remember, the organism on the right can only produce one type of gamete. They can only pass down one of the large chromosome and one of the small chromosomes, and the large chromosome will always have a small a and the small chromosome will always have a small b. So its gametes will always just be small a, small b. But for the organism that is heterozygous over here, it's going to be a little bit complicated. I'm going to color the chromosomes, okay? So you can see, I'm coloring it in blue and pink. So it can either pass down the large A, large B allele, as you can see here, or it can also pass down the small a, small b allele. We know this. But it can also pass down, due to random assortment in metaphase 1, it might also be able to produce small a, large B alleles, or it might also be able to produce large A, small b alleles over here. This is due to random or independent assortment. So when you are asked to write out the gametes, for the yellow round parent, it can produce large A, large B, large A, small b, small a, large B gametes, and small a, small b gametes due to random assortment. And for the green long one, it can only produce small a, small b gametes. In this case, do you have to do a Punnett square? I mean, if you want to, or if the question requires you to do a Punnett square, go for it. But it's quite simple to just do your line method. So that gamete will fuse with each other and it becomes large A, large B, small b. Second one over here, putting it in a different color, large A, small b, small b. Third one over here, small a, large B, small b. Fourth one will be small a, small b, small b. And the phenotype, the first one has one large A and one large B, so it will be yellow round. Second one is large A only with no large B, it's only small b, it will be yellow long. Third one is only recessive allele for A, but it has one large B allele, green and round. And the fourth one is green and long. The ratio of the offsprings in this case is 1 to 1 to 1 to 1, which means to say you have a 25% chance of producing each of those offsprings equally. It's not 9331, okay? 9331 is what happens when heterozygous is crossed with heterozygous. But if a heterozygous is crossed with homozygous recessive, in the case of unlinked genes, the ratio will usually be 1 to 1 to 1 to 1. Now, some students will still doubt me, so what I'm going to do is I am going to do another one. I'm going to make it heterozygous, which is yellow round, and heterozygous, yellow round. Remember their gametes? Their gametes can be the list of the gametes. I've listed it out there. Large A, large B, large A, small b, small a, large b, small a, small b. Whew, wow, really talking a lot here. And for the one on the right, same thing as well. In this case, you have no choice but to do a Punnett square. So when you do the Punnett square, I'm just writing it out here, the Punnett squares. I'm going to give you some time write out the genotypes first, okay, of the offsprings. What's the genotype of the offsprings going to be? Take your time, pause the video, and write it out. So these are the genotypes of the offsprings, and the genotypes of the offsprings, make sure your answer is also correct with mine, and then put in the phenotype. The phenotype, also pause the video if you have to, if you want to write out the phenotypes, okay? Remember, large A is yellow, small a is green, large B is round, small b is long, okay? As long as there's one large A and large B in the offspring, it will be yellow. So take your time, okay? Remember, if there's one dominant allele over the recessive allele, the dominant allele is always expressed. Pause the video, write out the phenotypes. You need to write out the phenotype for the color of the fruit and also the shape of the fruit, yeah? So once you're done writing it out, unpause and check your answer. So when you unpause and check your answer over here, these are the phenotypes, okay? Write out all the phenotypes. You're able to get yellow round, yellow long, green round, and green long. Check the ratio. The ratio for yellow round will be 9, yellow long will be 3, green round is 3, and green long is 1. 9, 3, 3, 1. When two heterozygous organisms are crossed, in the case of dihybrid unlinked inheritance. Okay, now I know it's quite a long video, but let's try the last one. Let's do a last one. Now, the last one over here is doing a test cross when it comes to dihybrid inheritance. Remember, test cross is what happens when you have an organism with an unknown genotype and you cross it with a homozygous recessive. So in my example here, the gene for body color, large B makes it blue, small b makes it red. Gene for body shape, large T makes it triangle, small t makes it square. Now, I have a parent here, and the parent's phenotype is blue triangle. We know that the parent will have one large B and one large T. We know that, but we do not know the empty spaces, because the empty spaces, the possible genotypes can be large B, large B, large T, large T, large B, small b, large T, small t, large B, large B, large T, small t, and large B, small b, large T, large T. These are the 4 possible genotypes, right? So which one is it? You might, sometimes in the exam, they may just tell you that the parent is a blue triangle. They may not tell you what's the actual genotype. To find out the actual genotype of this parent, you have to do a test cross where you breed the organism with a homozygous recessive. What's a homozygous recessive? So, homozygous recessive is small b, small b, small t, which is a red square. So, in this case here, we know that the red square parent will always produce a small b, small t gamete. That's fine. But if I'm just drawing out this organism's chromosomes here, as you can see, the blue parent here, it has B, question mark, we don't know the second allele, T, large T, question mark, we don't know the second allele as well for body shape. So we know that the gametes perhaps can be large B, large T, that's fine, but it can also be question mark, we don't know those two. It might also be due to random assortment, large B, question mark, or it might also be another one over here, question mark, large T. So the question mark is a little bit confusing. I'm going to substitute the question mark with a dash, okay, the empty space, so it's easier to see that. Okay, so these are the 4 possible gametes that it might be able to produce. So what we do is we cross them and we see the offspring's genotype and phenotype. So when we cross them together, we will get large b, large t, dash small b, dash small t. I like to put the recessive allele second. Okay, so we know that those 2 recessive alleles which I've highlighted will go there. And then the third one, large b, small b, we don't know that, dash small t, dash small b, large t, small t. So what the question will usually give you is, they will give you the offspring's phenotype. When you look at the offspring's phenotype in this case, no matter what, all the offsprings are blue triangles. So what does that tell you? If it's a blue triangle, for this one here, which I've highlighted, it has to have one large B to make it blue and one large T to make it a triangle. For that one over there, it has to have one large T to make it a triangle. And for that one over there, it needs to have one large B to make it blue. So obviously the unknown gametes are just large B, large T. So I've just basically put them all in. So this organism over here at the top is definitely just a homozygous dominant organism. There you go. Let's make it a little bit more complicated. Let's say that in this case over here, when they produce the offsprings in this situation, oh, this is interesting. It produced 50% blue triangles and 50% blue squares. See, to get a blue square, you need to have one large B, definitely, to make it blue. But to make it square, it has to be small t over there. Because only small t will make it squares. So the unknown gametes, which I've highlighted, so in this case over here, so I'm just highlighting again to show it to you how it works, okay? Right, so the parent here will have to have one large B and one small t. So it is homozygous for the body color gene, but it's heterozygous for the body shape gene. So it's a bit interesting here, which is large B, large T, small t. So that's the genotype of the parent right there. So you can use the test clause in dihybrid inheritance as well to determine the unknown genotype of the parents. Whew, we are done with this first part.